Welcome to Biotanemy.
Cancer signaling pathway. In the previous video, we learned how normal signal transduction pathways control cell growth, metabolism, and gene expression. However, when these signaling pathways become dysregulated, cells can grow uncontrollably, leading to cancer. Cancer arises due to mutations in genes that encode signaling proteins, including receptors, kinases, transcription factors, and tumor suppressors. These mutations convert normal signaling pathways into oncogenic pathways that promote uncontrolled proliferation, survival, and metastasis. In cancer cells, signaling pathways become permanently active, even without ligands. Tumor suppressor pathways are inactivated, and cells acquire self-sufficiency in growth signals and resistance to cell death. Major cancer signaling pathways are 1. Ras-RAF-MAPK or MAPK pathway. 2. PI3K AKT mTOR pathway. 3. JAK-STAT pathway. 4. WNT β-catenin pathway. 5. Notch signaling pathway. 6. Hedgehog pathway. 7. NF-kappa B pathway. 8. P53 tumor suppressor pathway. 9. TGF-β, SMAD pathway. 10. Hippo-YAP pathway. 1. Ras-RAF-MAPK or MAPK pathway. It is a receptor tyrosine kinase pathway. Please watch the video given in the icon above or in the description box to know more about the receptor tyrosine kinase pathway. The adapter protein GRB2 binds to the phosphorylated RTK via its SH2 domain. GRB2 recruits SOS, a guanine nucleotide exchange factor, to the membrane. SOS facilitates the exchange of GDP for GTP on RAS, a small GTP binding protein. RAS-GTP is now active and initiates the downstream kinase cascade. Active RAS-GTP recruits and activates RAF. RAF phosphorylates MEK. MEK1 or 2 are dual specificity kinases that phosphorylate ERK1 or 2, which enter the nucleus and phosphorylate transcription factors like ELK1, CFOS, and CMYC. This leads to changes in gene expression, promoting cell proliferation, differentiation, and survival. Commonly seen in melanoma, colorectal, and pancreatic cancers. Targeted therapies like BRAF inhibitors, MEK inhibitors are used. 2. PI3K AKT mTOR pathway is also a receptor tyrosine kinase pathway. Dimerization of RTKs activates the receptor's intracellular kinase domain, causing autophosphorylation on tyrosine residues. These phosphotyrosines act as docking sites for downstream signaling proteins. Phosphoinositide 3 kinase is recruited to the activated RTK via adapter proteins like IRS or P85 regulatory subunit. PI3K converts PIP2 in the membrane into PIP3. AKT or protein kinase B binds to PIP3 via its pH domain, localizing it to the membrane. AKT is phosphorylated and activated by PDK1, another kinase that also binds PIP3 at the membrane via its pH domain, and phosphorylates THR308 on AKT. mTORC2, recruited by PIP3 to the plasma membrane, will phosphorylate serine 473 on AKT. Active AKT acts on TSC2 of the TSC1/2 complex, which normally inhibits Reb, a small GTPase that activates mTORC1. mTORC1 executes downstream effects, stimulates protein synthesis via S6K1 and 4EBP1, and promotes cell growth, metabolism, and survival. Commonly seen in breast, lung, and prostate cancers. Targeted therapies like PI3K inhibitors, mTOR inhibitors are used. 3. JAK-STAT pathway. This is a type of cytokine signaling pathway, which we have already discussed in the previous video. Common in leukemia, lymphoma, and solid tumors. JAK inhibitors is used as targeted therapy for this pathway. 4. WNT β-catenin pathway. It is a GPCR-type signaling pathway. In the absence of WNT, β-catenin is kept low in the cytoplasm, since it is bound by a destruction complex consisting of APC, ASHIN, GSK3-beta, and CK1. GSK3-beta and CK1 phosphorylate beta-catenin, marking it for ubiquitination and proteasomal degradation. results in no beta-catenin in the nucleus, leading to WND target genes being off. WND ligand binds to a receptor complex on the cell surface consisting of frizzled receptor and coreceptor LRP5/6. Receptor activation recruits disheveled. DVL inhibits the destruction complex, leading beta-catenin to unphosphorylated and stabilizes in the cytoplasm, eventually translocates into the nucleus. Beta-catenin binds to TCF-LEF transcription factors and activates transcription of WND target genes, such as MYC, cyclin D1, and survivin, commonly seen in colorectal and liver cancers. Experimental WNT inhibitors are used to target beta-catenin pathways. 5. Notch signaling pathway. It is a juxtacrine signaling type pathway and is unique because the receptor itself acts as a transcription factor after being cleaved. Without ligand or in inactive state, the Notch receptor remains intact and inactive on the cell surface, and no signaling occurs in the nucleus. Ligands like jagged or delta-like on the neighboring cell bind to the Notch receptor on the target cell, triggering the atom, a metalloprotease, to cleave the extracellular domain of Notch. The remaining membrane-bound fragment is cleaved by γ-secretase. This releases the Notch intracellular domain into the cytoplasm. NICD moves into the nucleus and binds to the In many cancers, such as T-cell acute lymphoblastic leukemia, breast, lung, and pancreatic cancers, Notch signaling is overactivated or constitutively active, leading to uncontrolled proliferation, resistance to apoptosis, and maintenance of cancer stem cells. Targeted therapies used are γ-secretase inhibitors and monoclonal antibodies against Notch or its ligands.
The rest of the cancer signaling pathway, let's see in the next video. So, don't forget to subscribe. See you in the next video.
That's all for today's video. Thank you for listening. Hope all of you understand the concept, and for more queries and proposal, please comment down. See you all in a new topic.